It is my distinct honor and pleasure to introduce Christina Bergey to the speaker series. Dr. Bergey is an assistant professor in the Rutgers University Department of Genetics, studying how organisms adapt to their environment. Dr. Bergey earned her BA and PhD in anthropology at NYU, and after that was awarded a prestigious Kirstenstein National Research Service Award from the National Institutes of Health to pursue postdoctoral training in the lab of ASU alum PJ Perry at Penn State. As the co-author and author of dozens of scholarly publications, Professor Bergey studies complex polygenic traits for understanding past selection on modern medically relevant phenotypes, and has conducted research in collaboration with diverse human populations and within wild living primates and within malarial systems. So please join me in warmly welcoming Dr. Bergey to our seminar series. Thank you so much for being here.
Thank you so much, Katie. Thanks very much for the introduction. I'm really quite bummed that I can't be there in person to meet you all. And I know this year and last year has continued to be rough. So I hope this talk will serve as a slight distraction for the next 45 minutes or so. I'll be talking today first about the work I began in Penn State in ASU alum PJ Perry's lab, working on the genomics of rainforest hunter-gatherers, and which I'm now continuing ever since I began my own lab at Rutgers right before the pandemic started. So I should start by confessing, you heard my backstory, that when I was a proper primatologist back in my undergrad and grad school days, I confess that I always thought that humans were very boring. We just don't differ from one another very much. So if you think back to the last time you were in a crowd of people, it may have to be your last like COVID anxiety nightmare. If you were to scan that crowd, you would notice that one of the ways we do differ from one another is height. So these Yukon kids here are standing uncomfortably close to one another to create a living histogram of height, with the women in white and the men in navy. And you can see that height varies a great deal. You're probably thinking from my imposing Zoom presence and confidence, I'm at least 6'2", 6'3", but in reality, I'm down here on the left. So besides being an obviously salient feature in social interactions, sorry, I'm just smiling because the only person I see laughing at my jokes is Kenny, which is wonderful to have. So besides being an obviously salient feature for social interactions that are not Zoom-based, height is a fundamental trait that has a lot of influence on health. So as a short person, I will have a higher risk of heart disease, for instance. I'm more likely to be in a lower socioeconomic status. And it's not nothing. One recent study found that for every 2.5 inches of height, so every standard deviation you are taller, you have an increase of, it comes with a reduction, 13.5% reduction in heart disease risk, and almost $4,000 more in annual household income. I remember reading those studies and being very angry on behalf of all shorter people. But height not only differs within populations, but also between populations. So the average male in our Yukon histogram is 5'10", or about 177 centimeters tall. They're a bit taller in the Netherlands. Among the shortest populations are the Botswa from Africa and the Onge from Asia, where males are on average less than 155 centimeters or 5'1". And if you look at human height across the world, you'll find that these shortest groups are hunter-gatherer populations in the tropical rainforest belt, which is shown on this map in green. And these groups have historically and sometimes racistly been lumped together as pygmies, which I say with air quotes. And rainforests are difficult environments for human habitation. They're hot and humid, they have dense vegetation, and there's not much food or protein available to humans. There's also plenty of parasites and disease. So to survive in the rainforest, hunter-gatherer populations from South America, Africa, and Asia have all independently and convergently evolved small body size, making it, in my opinion, one of the coolest examples of convergent evolution, full stop, not just in humans. And since these similar ecological conditions have led to convergent biological adaptations, so this convergence then presents us a natural laboratory to better understand how evolution works. We can learn a lot about how evolution works by examining how humans have inhabited these extreme environments. So well-known examples to students of human diversity include places like the Arctic and Antarctic, high altitude hypoxic environments in places like Tibet and Ethiopia and the Andes, and then even physiological adaptations to diving, to marine subsistence. Of these, a tropical rainforest may appear to be the most forgiving, but rainforests are in actuality a rough place for people to live. And there's some conjecture as to why people living in the rainforest have independently evolved this small body size in parallel. And because, as I said, because humans have moved into rainforests independently all over the world, we can then use this this parallel colonization of the rainforest to test theories about how evolution works and whether natural selection targets the same genes or pathways under repeated trials. These people here are the Mabuti from the Congo forest. I was lucky enough to travel in DR Congo with Therese Hart, who took the photo in the upper right when I was a grad student. Oops, sorry. So various adaptive hypotheses have been put forth to explain the convergent small body size of worldwide rainforest hunter-gatherers. These theories include lack of food or protein in rainforests, high mortality from disease that incentivizes early maturation and reproduction, and even somewhat absurd hypotheses. The person in the lower right is meant to remind me, like a decrease of risk of falling out of trees for small people. Although as a small person, I've fallen out of the tree, and I don't think being small helped me all that much. These hypotheses about selection concern the ultimate evolutionary cause of the small body size phenotype, and they're still hotly debated among anthropologists, but we also don't know much about the underlying proximate mechanisms. So for this first part of my talk, I want to discuss our population genomic investigation using diverse human populations in the rainforest to try and understand the proximate genetic underpinnings of the short stature and small body size in rainforest hunter-gatherers. So for this project, we wanted to find out the genetic underpinnings of the small body size trait in rainforest hunter-gatherers. RHGs is rainforest hunter-gatherers because I've been typing it for years and it takes long to type. So with these data in hand, we could then test if the convergence in the trait was due to parallel changes on the same genetic pathway, or if evolution instead took a separate path each time, targeting different genes and pathways. So to explore how selection may have influenced the genomes of rainforest hunter-gatherers, we used two data sets. The African data set was generated by our group, included the 50 individuals from the Batwa of Uganda, and the Asian data set was published by Mandel et al. And included 10 Andamanese Islanders individuals from islands off the coast of India. And then for each population, we also had comparative agriculturalists, so 50 Bachiga of Uganda for the African data set, and 10 Uttar Pradesh Brahmins from mainland India as comparisons for the Andamanese Islander data set. So for this first part of the study, we looked only at protein coding regions, so exonic SNPs from the exomes of these individuals. And the goal was to find regions of the genome that might be under natural selection and therefore underlying the adaptive evolution of the small body size phenotype. So to do this, I used a very simple statistic in population genetics. The population branch statistic is called PBS. It's based on FST, and it's really just a measure of how different two groups are in their allele frequency at a given SNP. I feel like I'm tipping my hand to the non-geneticists in the audience by revealing that so much of genomics is just very basic arithmetic on a grand scale. So we used this population branch statistic, PBS, to find places in the genome where there's evidence of selection on both the African Batwa rainforest hunter-gatherers and the Asian Andamanese rainforest hunter-gatherer branches, the two groups with the short stature, to the exclusion of the nearby agriculturalist populations. And then we polarized the change with outgroups to know if the adaptation occurred on the branch leading to the hunter-gatherers or the agriculturalist comparison. So using these PBS values, the selection metric, I computed a selection index, which you can interpret as akin to a p-value for all SNPs and genes. So the non-synonymous SNP, or the single varying base pair with the strongest evidence for convergence in the two rainforest hunter-gatherer groups was found in this gene, FIG4. It's very hard to Google because it's FIG4 is how people, it's often found in papers that have more than 3 figures. So it's kind of a pain to figure out what's going on. But if you persist, you'll end up finding that when FIG4 is disrupted in mice, it results in a phenotype of small but proportional body size, which is precisely what we see in rainforest hunter-gatherers. So this is definitely something we want to look into further. However, this is just one gene. Height is the textbook example of a polygenic trait or a complex trait, meaning it's influenced by many genes spread throughout the genome. So we next moved beyond the single SNP and gene results to look at pathways of genes with related functions. So we wanted to know whether the genes under selection were more likely to be in some functional category. And this is a polygenic question by definition, since it focuses on classes of genes united by their shared function. There's two general classes of tests that can be used to test this hypothesis. The first are outlier-based tests, answering the question, do these pathways have a ton of extreme outlier values of genes under selection? However, we knew that we were looking at height, which is polygenic, and we expected to find kind of tiny concerted changes in allele frequency across many genes. And these can be better found using a shift-based test. So that's what I'll focus on today. You can read the paper for details, but we used a Kolmogorov-Smirnov test to find pathways that showed a shift in the distribution of the selection metric relative to the background genome-wide distribution. And we did these tests on functional categories called gene ontology, or GO, biological processes, and molecular functions. So this plot shows each functional category's p-values for the shift-based test for selection, plotted on a log scale. So for the Andamanese Islanders, the Asian population of hunter-gatherers, is on the y-axis. I don't think I have a cursor, so I'll paint you a word picture. And then the African Batwa are on the x-axis. So categories at the upper left of this plot have signatures of polygenic selection in the Andamanese Islanders. Blue points on the lower right have signatures, or are functions that have signatures of selection in the African rainforest hunter-gatherers, the Batwa. And then what's kind of nifty is, to me, are the points in orange in the middle. So these orange are functional categories with signatures of selection in both the Asian and African rainforest hunter-gatherer populations. Excuse me. And here the point size is proportional to the number of genes in each functional category. So for the convergent functions, sets of genes, the top molecular function was growth factor binding, and the top biological process was organ growth. So this suggests that evolution has convergently acted on these growth-related pathways when African and Asian rainforest hunter-gatherers became smaller, which I was very excited to see. So we kind of expected that, not that you're supposed to kind of be looking for something in particular when you do such blind surveys, but we thought we'd find growth-related pathways at the top of these convergent lists. There was unexpectedly, however, at the top of both our convergent and our population-specific results, a bunch of heart-related pathways. So for instance, up in the upper right, cardiocyte differentiation in the Andamanese Islanders, cardiac ventricle development in the lower right for the Batwa, and in the middle, in orange in both, cardiac muscle tissue development. I should note, in contrast, that when this analysis was repeated on the agriculturalist populations, no growth or heart-related functional annotations were observed with significant convergent shifts in both populations. Meaning this is really something that's specific to hunter-gatherers. It's not really a general evolutionary pattern. Oh, I included a slide. This is what I just said. There's no growth or heart-related convergent pathways in the agriculturalists. So these signatures of heart-related of selection on heart-related pathways didn't really make a ton of sense until I thought back to my short person increased risk of heart disease. So these papers here show that the heart is connected with height in general and growth hormone, the main hormonal regulator of growth in particular. So for example, if you read these papers, short people have an increased risk of atherosclerosis. Patients with growth hormone deficiency have worse heart function that's rescued if you administer to them a growth hormone supplement. So based on their short stature and the altered growth hormone response we predicted, we would expect that rainforest hunter-gatherers would have higher rates of cardiovascular disease symptoms, including things like elevated systolic and pulse blood pressure and arterial stiffening. However, in contrast, what few data do exist suggests that hunter-gatherer populations have better or similar blood pressure and arterial stiffness, especially that those benefits seem to extend into aging. Obviously, there's a lot of environmental factors that I'm largely glossing over, but I do kind of have this theory based on the selection signatures that we've found that if rainforest hunter-gatherers became short by evolving overall growth hormone subresponsiveness, then this may have necessitated compensatory changes in the cardiac pathways in which growth hormone also plays an essential role. So this work was published. If you're curious, you can read more about kind of our speculation about what these means and why we were kind of excited to see the heart results show there. And we succeeded, I think, in our original effort, which was to partition the variation we see underpinning the short stature into that which is convergent but in parallel between the populations, as well as some that seems to be specific to the African or Asian hunter-gatherer populations as well. So unfortunately, though, this investigation was based on exomes, or only the protein coding regions of the genome. So there's always a little bit of like a missing piece. Nonetheless, with this exome data, we were able to get some insight into the proximate genetic mechanisms that may be underlying the small body size phenotype. However, there were still some unanswered questions. So namely, we wanted to know, is this selection that we're seeing or that we're intuiting on height and heart-related genes, is it ongoing, or is it something that's kind of occurred in the past? And then secondarily, can we produce any insight into these larger adaptive hypotheses? Why is it the ultimate evolutionary reason? Why is it that people, when they go into rainforests, become smaller? And then finally, this one was kind of suggested by our previous study. This heart selection that we're seeing, is it just a byproduct of the selection on height? Are these two traits pleiotropically linked, or is it independent? So we since this first study built on our previous work by sequencing the whole genomes for about 80 Batwa individuals to follow up and try to hopefully answer these questions a bit more satisfactorily. So on the whole, we did confirm our previous results, so I won't subject you to more redundant graphs. However, there's a few additional insights that the whole genome analyses afford that I'll talk about now. So the first pathway that the whole genome analysis opens up is the ability to look at how ancestry varies across the genome for some insight into recent selection. So the Botswana rainforest hunter-gatherers have a long history of interaction with and intermarriage with nearby Bantu agriculturalists. You can see in this plot, I wish I had a laser pointer. You can see that in this plot, the amount of Botswana, the Botswana ancestry component is shown in yellow, and the Bachiga Bantu agriculturalist ancestry component is shown in blue. Each vertical line is a person, so this analysis partitions an individual's ancestry into a hunter-gatherer and agriculturalist component, done by ASU's own George Perry, I should add. And you can see that there's some Botswana individuals who have similar levels of agriculturalist genetic ancestry as the blue, the folks on the right, the Bachiga agriculturalists. And there's a lot of variation in the amount of hunter-gatherer versus agriculturalist ancestry in each individual. And we know from this prior work by ASU's own George Perry that this overall ancestry correlates with height in the Botswana males and females, shown in the scatter plots in the lower right of showing height on the y-axis and proportion of ancestry on the x-axis for males and females separately. What we can do with the whole genome sequencing is we can do a more granular analysis and find regions of the genome where these admixed individuals have more or less agriculturalist ancestry. Regions with more or less agriculturalist ancestry may be those that are where selection has either favored introgressed agriculturalist ancestry, or conversely has selected against it and removed it post-introgression. So there's kind of a imperfect proxy of recent selection, post-introgression selection. So this plot shows the amount of agriculturalist or conversely hunter-gatherer ancestry along each of the 22 chromosomes. Red regions have higher levels of Bantu agriculturalist ancestry in the population, while yellow regions have relatively more hunter-gatherer ancestry. And this is really focused on those individuals, I should say, Bantu individuals, or sorry, Botswana individuals with some evidence of admixture. So as an example, if we zoom into chromosome 22 down at the very bottom, you can see that this genomic region with lower, you can see there's this genomic region where there's a dip and there's a much lower agricultural ancestry, or conversely higher hunter-gatherer ancestry. And this gene is PRR5, is kind of right in that neighborhood. PRR5 is a subunit of mTORC2, which regulates cell growth and survival in response to hormonal signals. And so mTORC2 is activated by growth factors, and it seems to be nutrient insensitive. So it's somewhat independent from nutrient intake. And so if you then expand from this and look across the entire genome and find the more yellow bits with more hunter-gatherer ancestry, you can ask what type of genes are in these yellow regions. What regions of the genome does it seem like selection is preferring or selecting the hunter-gatherer portion? If you do so, maybe somewhat surprisingly, these regions with higher Botswana rainforest hunter ancestry and admixed individuals are enriched for growth functions as well as circulatory system involvement. And there's some heart regions a little bit later on in this table. So these are the enriched functions showing the number of significant genes, how many we would expect, and the statistical test for whether that's more than we would have expected by random chance. So this builds upon our recent work by bringing insight into recent selection. And this may indicate that there's continuing recent selection related to body size and heart development in admixed individuals, and that selection in this case may be favoring the Botswana hunter-gatherer forms of these loci. And this is fairly preliminary. We're following up on this exciting result right now. So selection targeting heart and height-related loci seems to be ongoing, or at least recent since the admixture with Bantu farmers. So our next kind of outstanding question was whether we could learn anything about the adaptive hypotheses concerning the ultimate evolutionary reasons for the small body size phenotype. So get away from just the, this gene seems to be involved in growth and move to the larger picture, why is it that people become small when they move into rainforests? So just to remind you, here are some photos from before when we discussed the various adaptive hypotheses that have been proposed to explain the convergent small body size in worldwide rainforest hunter-gatherers. The two theories that to me hold the most water, include lack of food or protein in rainforest, accessible food or protein, as well as the alternative hypothesis that it's a life history explanation that high mortality from disease incentivizes early maturation and reproduction. And so if you're gonna reproduce quickly, you don't want to, I'm personifying it, but you don't invest in growth, you invest in making it to maturity earlier. So with using the whole genome sequencing data, we wanted to know if there were any signatures of selection on genes that might be tied to metabolism on the one hand, or alternatively, life history traits. It's a little bit of a false dichotomy though. These aren't really mutually exclusive hypotheses. So to look for loci with insight that might provide insight into these adaptive hypotheses, we did a similar scan for selection as before. And there's a lot of stuff crammed onto this plot, but I'll explain it. So this metric shows, it's a composite selection score. So essentially it's a summary of multiple disparate tests for signatures of selection. So these include metrics based on allele frequency, like the population branch statistic we used in the exome data set. And then because we had the whole genome data, we could also phase the genomes into haplotypes and use haplotype length, which is a powerful tool for detecting selection. So a bunch of these metrics are kind of with overlapping pros and cons are kind of combined together into this composite score. And higher scores, which are also shown by being colored yellow or red, are regions with the strongest evidence of selection. So based on this composite selection statistic, this region right here has the strongest evidence of selection in the Botswana rainforest hunter-gatherers. And if you zoom in, you'll see that the peak in the statistic aligns with two genes, DHCR7 or 7 dehydrocholesterol reductase, and NADSYN1, NAD synthase 1, which both of these are connected to metabolism. They're very close by. So this locus has been shown to influence circulating vitamin D levels, which itself has been linked to various diseases, including cardiovascular disease. And this region is of interest due to the selection on metabolic traits that is hypothesized to accompany human habitation of rainforests. So this 7-DHCR7, for instance, DHCR7, for instance, catalyzes the first step of the biosynthesis of cholesterol, which is the precursor to vitamin D. And it might regulate the switch between cholesterol and vitamin D synthesis. So this critical role in metabolism is reflected in the fact that if the genes are disrupted, or when the gene is disrupted, abnormal growth occurs. So this might be some evidence that there is selection related to metabolism that coincided with human habitation of the rainforest. So that was just one gene. And most of these traits of interest are complex or polygenic. So we can additionally look at this composite selection statistic in the vicinity of a loci that are known to be associated with various traits that are tied to our adaptive hypotheses. So first up, you can look across the genome at places known to influence particular traits like height and ask whether there's indications of natural selection in these regions, more than would be expected by chance. So this plot shows the p-value plotted on a negative log scale. So farther to the right means stronger evidence of selection. It summarizes the test for whether these regions linked to these traits are selected in the Batwa hunter-gatherers. And indeed, maybe not unsurprisingly, we see in the Batwa hunter-gatherers evidence for selection on regions linked to, both standing and sitting height. And then looking at these next traits that are kind of tied to these adaptive hypotheses, we also see selection on metabolism-associated traits like body mass index, basal metabolic rate, as well as traits linked to life history or maturation, like menarche, or the timing of the age at the first period. And then just because of the prior exome-related results, I've also added a bunch of traits related to heart disease and associated phenotypes. And you'll see that all of them on the whole, systolic and diastolic blood pressure, triglycerides, and overall coronary artery disease are significantly, have significant signatures of selection in the Batwa. The one that doesn't meet the criterion for significance is LDL levels. That doesn't seem to be selected in these hunter-gatherer population. So this is further evidence that heart functions may have been influenced by selection in the Batwa. However, the selection for height is so intense and pervasive. It's been described as omnigenic because it impacts the entire genome. So maybe this selection that we're seeing on all these different traits could just be due to nearby selection related to height impacting nearby loci. So I'll walk you through this by just focusing on these two metabolism traits at the moment and kind of talk through the tests. So if you remove from the analysis all genomic regions linked to height, you can then test for selection in the genomic regions that remain to see if the signature you're seeing is just a byproduct of height. So for instance, in the top arrow, BMI, which is significant when you include the height associated regions, moves far to the left when you take away those height associated regions, but there's still significant signatures of selection on BMI when height-related loci are removed. So this suggests some independence between BMI and height. However, a BMR, basal metabolic rate, is no longer significant when height loci are removed, suggesting that selection on basal metabolic rate may be linked to or a byproduct of height selection. We can do this for all of our traits. So for instance, menarche is barely non-significant when height-related loci are removed. And then for the heart-related traits, systolic and diastolic pressure are independent of height significant, with or without the height-related loci. Triglycerides, however, and the coronary artery disease, both are no longer, from being fairly significant to not at all when height-related loci are removed. So this suggests that the heart selection we're seeing in our analyses may very well be a byproduct of selection on height, and that selection may be targeting the regions that link heart disease and height through epistasis or through the shared genetic architecture. This makes sense given what we see in Europeans, where heart disease-related loci and height have shared genetic architecture. They're linked via pleiotropic connections between the various genes. And there's one more nuanced analysis that we can do as well, and this may be pushing data a little farther than they have any right to be going. But one thing we can do, rather than just look for selection in windows containing loci linked to traits, we can also compute what are called polygenic scores or polygenic risk scores. So in this little cartoon, these loci have been linked to height through outside GWAS analyses, association analyses, and having one or two copies of these variants indicated by the purple arrows, is predicted to either increase or decrease your height depending on what has been found in the previous association analyses. So overall, this imaginary person, if you sum up all these values, should be 0.75 centimeters taller because he or she has this particular set of variants. The same thing can be done for a risk of heart disease. You can sum up the variants that increase or decrease your odds of coronary artery disease, say, and you can compute a person's polygenic risk score for heart disease. So to test if two traits are linked by common genetic architecture, there's this neat little trick you can do. So you can predict a person's predict a person's trait based on DNA alone for the one trait. So for this case, we can predict a Baka individual's risk for a heart attack based on just their genome. Then you can test whether this predicted genetic risk is correlated to the second trait, in this case, height. So if the genotypic prediction of a heart disease correlates with the actual height phenotype, then you know that those two traits are epistatically linked and that they share genetic architecture. And that may very well be the case here. It's a very weak correlation, as you may expect, given our sample size and the massive environmental impacts on things like height. But for males, at least, taller folks have a lower risk of heart disease, as is the case in European studies. There may be a sex interaction here with females not having the predicted pattern, but like I said, these are weak, and I don't have a ton of confidence in these results at the moment, so please take this with a massive grain of salt. One major caveat, and why I'm being a little bit cagey here, is that these GWAS analyses that these polygenic scores are based on are done primarily in European populations. So translating that insight into Batwa African populations is very fraught, to say the least. But in any case, such work can begin to allow us to examine if heart and height are linked in the Batwa by directly testing it the same way that they have been found to be linked in Europeans. So from our whole genome sequencing data, we can then fill in a bit of the answers to our earlier questions. So selection on height, selection on heart disease, and some but not all components of phenotypes related to heart disease disappear when height-related regions are removed, suggesting epistatic links between height and heart disease traits. And then we also find additional evidence of selection on both metabolism and life history-related traits, and these are largely independent from body size-based selection. One of the frustrating things about population genetics, however, is that you can never be completely sure if the signatures of selection you're seeing are actually tied to a certain trait. So these genes under selection have been linked to growth and cardiac development in mice, as well as human populations, primarily Europeans, but we can never, at the moment at least, be completely sure about their effects in these rainforest hunter-gatherer populations. And confirming the link between genotype and phenotype requires experimental manipulation. So I'm going to very briefly talk to you about some ongoing work we were doing using functional genomics to uncover the genetic underpinnings of small body size phenotype in the Batwa rainforest hunter-gatherers. So the main growth pathway in postnatal growth is the GH-IGF-1 pathway, named for its two key players, GH or growth hormone shown here in green, and IGF-1 or insulin-like growth factor in gold. This is a really simplified cartoon of how the GH-IGF-1 pathway leads to skeletal growth. So the original straightforward story is that GH from the pituitary gland circulates and causes the liver to produce IGF-1, which in turn signals cell proliferation at the growth plate of say, long bones. Previous studies in rainforest hunter-gatherers have found that circulating growth hormone levels were similar to agriculturalists. However, the response to growth hormone was muted, and the IGF-1 serum levels were low, especially during the puberty growth spurt when IGF-1 typically spikes. So taken together, these studies suggest that rainforest hunter-gatherers have typical growth hormone levels, but the downstream effects of growth hormone are somehow muted, likely causing their short stature. So we've done a few efforts to understand the genetic cause of this differing response to growth factors. However, manipulating growth hormone levels in living humans, like they did in the 1960s, is horribly unethical. So instead, we've been doing the experimental test of response to growth factors in vitro with various cell lines. I won't talk to you too much about them. This is supposed to be the triumphant summary of these results, but COVID has unfortunately delayed this a good bit. But in a nutshell, typically it involves taking cell lines, immortalized cell lines, growing them up in flasks and exposing them to growth hormone, and then sequencing the transcriptome to get at gene expression data to measure all genes at once to try and investigate the genetic architecture of this trait. One of the major issues with using immune cells to study stature is that immune cells don't really have much to do with stature. So immune cells are very easy to get and manipulate, and they're useful models for telling us a ton about overall pathway differences. But it would be so much cooler to have, say, cartilage cells to look at long bone growth, or liver cells to look at how growth hormone induces IGF-1 production. But obviously, speaking of unethical research, invasive biopsies in a healthy person, let alone entire population, is just not feasible. So we've been doing a lot of this work and more using the new techniques to create what are called induced pluripotent stem cells, or iPSCs. So with these methods, we're taking primary somatic cells and de-differentiating them back to a stem cell state that has the capacity to differentiate into any other cell type. So we have the power to essentially rewind the developmental clock and then guide the cell into a new fate. We've done this. We have created iPSCs for the Batwa and Bachika agriculturalist cells. And we have all indications that they're good and have pluripotency as shown by these fluorometric assays. With these, one of the cell types that I'm very keen on is hepatocytes, or liver cells, because rainforest hunter-gatherers have typical growth hormone levels but low circulating IGF-1. So the liver might be the key place where this key endocrinological regulator of small body size. So anyway, this is an ad for upcoming research. And I'm a little bummed I can't talk more about it, but it's just not ready for prime time, unfortunately, because of the pandemic. Okay, so I want to end with a very quick departure about intestinal worms. So I hope I've overall convinced you about how genomics can be used to understand the proximate as well as the ultimate reasons for rainforest hunter-gatherer small body size. And I want to end my talk by discussing a little bit about one component of the adaptive theory related to life history timing. So the theory goes that because rainforests have so much selection pressure due to the high pathogen load and the high parasite burden, this incentivizes early maturation and reproduction. So you don't get tall, you don't invest in increased body size. You just go as quickly as possible to having kids, because the concern is you won't have that much long of a window because this it's a rough environment to live in terms of pathogens and parasites. If you're interested in the pathogen side of things, this is a plug for this pub led by Janelle Harrison, and on which I played a small role. And it did the work to expose these same cell lines to bacteria and virus simulators, and they found increased divergence between the hunter-gatherer and agriculturalists in the response to viruses, not so much for bacteria, somewhat surprisingly, but even more surprisingly, they found that the signatures of positive selection were disproportionately observed in the hunter-gatherer groups, a counter to what we might think, given our mental model, or at least my mental model, that agriculture was what led to a shift in the pathogen-related selective pressure. So check that out for the pathogen story. The little bit I have to talk about today is focused on the parasite response. So this is me with a bear tapeworm. This is like the most disgusting thing I've ever done for science. We had someone in Alaska shipped us 2 boxes of bear intestines to try and paw through them and get worms out. But unfortunately, it was the middle of summer, and the FedEx person erroneously told her that it was illegal to ship ice packs. She'd just have to ship them at room temperature. So that meant a box full of bear guts came to the lab with my name on it for me to go through and get tapeworms. So anyway, that was how everyone in the entire department just hated me for like 2 weeks during one hot summer in Pennsylvania. So anyway, that was just an aside because I wanted to complain about it because the trauma still has not passed. So this is not my work, but studies of hunter-gatherers in Cameroon, for instance, have found that rainforest hunter-gatherers, shown here in the red bars, have very high prevalence of intestinal parasites. So parasite load is indeed higher in the forest. Perhaps because of this ecological difference between the populations, agriculturalists, hunter-gatherers, or pastoralists, we hypothesize that natural selection may have targeted Helminth response genes in rainforest hunter-gatherers, going along with that theory about the ultimate causes for small body size. And luckily, we had a data set that we could use to generate a set of genes to test this hypothesis. So this work was done with my student, Amber Trujillo, seen here smiling next to intestinal parasite, a Trichuris whipworm. So we took RNA sequencing data from the feces of gorillas and Baka rainforest hunter-gatherers living sympatrically in Cameroon. It's not these folks, these are gorillas, but this is a selfie from Virunga that I just really thought was fantastic, so I included it. So using this RNA data, we were able to get a proxy for parasite load by comparing the amounts of parasite reads between the samples and between the species. And because we had RNA from feces, which is actually coming from the intestinal cells, we also had gene expression data from the host, human or gorilla. So we could find genes that were differing in response to worms, either by species or just overall responded to worm infection. And this kind of dual transcriptomic method in which you assay both the host and the parasite at once has been a cornerstone of a few projects we have going on in my little baby lab at the moment. So I'm going to focus on this Trichuris whipworms for a second and just focus on the human response. So shown here are the functional categories of genes that are overrepresented for genes that differ by species in response to Trichuris. That differ in humans in response to one of these worms, this whipworm. So genes that respond to these whipworms, for instance, are significantly more likely than expected by chance to have arachidonic acid binding functions, for instance. This is kind of cool because arachidonic acid is an important inflammatory mediator. And one cool finding about arachidonic acid and helminths comes from this guy, this little water rat, who's resistant to schistosomes, or blood flukes. And a researcher found that with repeated infection, they accumulate arachidonic acid in their liver and in that way become resistant to the blood flukes. So this arachidonic acid is a potent natural worm killer. So there's this intriguing possibility that humans are producing it in response to helminths, but that's neither here nor there. I just thought that was kind of nifty. Anyway, putting aside the water rats, our main goal for this was to see if there was differential selection in the hunter-gatherer population on genes involved in parasite response. And this is linked to that overall adaptive hypothesis that high mortality due to the high pathogen load and the high parasite burden of rainforests incentivizes this early maturation and reproduction at the expense of growth. So if this is the case, then one prerequisite for this would be that there has to be a selective pressure from parasites seen in the hunter-gatherers, but not the agriculturalists. And so with this data in hand, we could finally test this longstanding hypothesis that intestinal worm or helminth parasite response genes have been under differential selection in agriculturalists versus hunter-gatherers, rainforest hunter-gatherers. So to do so, we can simply compare the proportion of selected genes in worm response genes compared to all in the set of worm response genes compared to all of the genes. And so I found that for ankylostoma hookworms and Ascaris roundworms, which I have some experience with, worm response genes are significantly more likely to be selected in hunter-gatherers. And there's nothing at all significant when we do this in the agricultural population, meaning, so in that case, worm response genes are no different than the other genes, meaning there's selection on helminth response genes that's specific to the rainforest. So with that, just to sum things up, I know it's a kind of a little bit of a wide-ranging talk, but there's so many cool things you can do with this genome data set to look at selection and kind of tie it to these hypotheses that various folks have had for human habitation of the rainforest. So using the exome and the whole genome data set, we looked first at, or using the exome data set, we looked first at whether or not African and Asian rainforest hunter-gatherers have evolved small body size using the same genetic mechanism, and we found a mixture of shared and as well as population specific genes and pathways involved in both. We also found this kind of somewhat intriguing possibility that there is selection on genes related to heart development in addition to height. And through the work with the whole genome sequencing, we were able to kind of looked at that in a bit more detail and found that in some instances, selection on heart-related traits are partially a byproduct of height selection. They're linked as they have been found to be in European and Asian populations. And then finally, nothing's definitive, but we did find some selection on genes related to traits that tie into these larger adaptive hypotheses for ultimate evolutionary explanations for why small body size evolved in the rainforest hunter-gatherers. So these are things like metabolism, but also maturation timing and parasite response genes in the hunter-gatherers. So with that, this is a very small subset of the people that I should acknowledge, but in particular, I would like to thank the Batwa and Bachiga participant communities. I was lucky enough to go back and visit them a few times to present a similar talk to this one, and I'll be eager to do that in the future. That's me and presenting some of our work a year or two ago. I'd also like to thank PJ Perry, ASU's own PJ Perry, who was the PI at Penn State when I began this work and has graciously allowed us to kind of continue the collaboration. And I'd like to thank my lab group as well, especially the PhD student, Amber Trujillo, who's did a lot of the worm response work so far. So thank you very much for letting me tell you about some of this work, and I'll be happy to take any questions you may have. Thank you.
All right, fantastic. Thank you so much. This is great. You're getting lots of accolades in the chat window.
Thank you.
Yep. And so let's open the floor up to any questions from our audience.
I like the response that's nasty and you're brave, which I assume is in response to digging through the bear intestines.
That was
disgusting.
That was exactly it. Your description transported us to what that would have smelled like. So yeah. Thank you.
Yeah, you're welcome.
Yeah. So I guess I'll start out with a question, which is where are you most excited to start putting efforts post-pandemic? Because obviously you have so much of a broad research program, lots of really neat clues, lots of new directions, and so what are you chomping at the bit to do most next?
Yeah, that's a good question. Yeah, besides just going to a restaurant and sitting down within 6 feet of some other human, work in terms of work-related stuff. Yeah. The one that was quite of a bummer to kind of get delayed a little bit was the efforts to create these hepatocyte-like cells, these liver-like cells, because I really think they hold the clue to really starting to, for the first time in a long time, or first time forever, disentangle the genetic underpinnings of this phenotype. It's such a cool story in human evolution that folks were able to live in this rough environment. And there's so a lot of speculation about how it's even possible that such a thing could happen. But I think that'll really start to get down to the phenotypic level, the genetic level, what's going on. And it's finally gotten to the point where such methods are available to evolutionary anthropology, evolutionary genomics, where you can really start to, they're feasible enough for folks like me to adopt them and use them. So besides the hepatocyte-like work, which is going so far so good, the one I'm very keen on, and it's a bit more of a shot in the dark. Like if I asked the NIH for money for it, they would say no, and they should. But is to create to use these stem cells to create cardiomyocytes, so tiny cardiac muscles, heart muscles, which actually will beat. They actually will spontaneously start beating in the dish, which is, to be honest, probably one of that alone is enough reason to do it. But I think that will be something that could be very, very interesting to see how these signatures of selection that are just kind of pointing towards in the general direction of heart muscle and heart development could be the way we could really see if what we think might be going on has actual real phenotypic consequences.
Fantastic. Thank you. And
yeah, of course.
Ben, you've got hands up now. So Ben Trumbull has a question, Professor Ben Trumbull.
Sure thing.
Hi, great talk. Thanks so much.
Thank you.
So I had a question. So you mentioned earlier that, risk score doesn't necessarily mean heart disease, particularly since they're largely based on European samples.
Right.
So do you see any heart disease in the population?
Yes. So it's a great question. Unfortunately, for the Botswana and to a lesser extent, unfortunately for me, until very recently, they didn't have the kind of medical care where that would even be evident. So that has changed with a regional hospital that's actually a fantastic hospital that's grown up with the Botswana, and that's something we're hoping to look at. So we're hoping to go to where the Botswana live with portable machinery to look for to essentially do a cardiac workup, EKGs and the like. But as of right now, there's no data exists on prevalence of heart disease as far as I know. There's also my cardiologist friends collaborators gave me grief for this before, but I should distinguish that there's the genetics of cardiac disease and the genetics of heart development are often quite development disorders are often quite separate, and it's an unfortunate case that quite often if kids have high rates of heart disease, they probably unfortunately just die as newborns, and the cause is never known. So one other effort we're doing is to go and do this phenotyping in Uganda. We're hoping to get it started after the pandemic, to be able to start to see if what the prevalence is, and potentially how that has changed with the adoption of a less traditional diet and subsistence strategy. But yeah, it's a good question, and unfortunately, the short answer is we don't have much data.
Well, I think it's especially interesting, actually, because so other hunter-gatherer and forager horticultural populations show relatively low rates of heart disease.
Yes.
Since organs scale to height, you could imagine that if individuals are shorter, they're going to have slightly smaller hearts, potentially. And that would mean that you could have, even with a lower lipid load, you could have the hardening of arteries and closing off of arteries earlier than, say, an individual that was taller.
Yeah, precisely. I think a lot of it comes down to just the arterial diameter. Excuse me. But they haven't fully nailed down what precisely it is about height that precisely links it. It's not fully known, although that's one major component of it, the heart disease height risk. So yeah, it'd be fascinating, I think, to see that, especially given the weird signature of selection we see, where there may be some interesting phenotypes that are particular to the Botswana hunter-gatherers. But yeah, it's a good question. Thank you.
Fantastic. Thank you. And now Alexis also has their hand up.
Yeah, hi, thank you. So one of the curiosities that popped into my mind during this was because short stature in the rainforest populations is linked to early maturation, do these populations tend to have shorter lifespans? I don't know if this is something that you looked into, but it was just something that I was curious about.
Yeah, no, it's a great question. So a lot of I probably played a little fast and loose with talking about the early maturation life history hypothesis, because at least in West Africa, I believe a lot of information on longitudinal growth has, to a certain extent, taken the wind out of the sails in that hypothesis, that it doesn't seem like there's this rapid maturation. I don't know if it's a done deal. It still seems to be a potentially valid hypothesis, at least theoretically. But what limited data we have does kind of call that into question. That being said, the only there's an awful dearth of even basic data on some of these populations made vulnerable by government and societal discrimination, of course. And I've seen some alarming stats about the life expectancy for Botswana individuals being like only 38 years, obviously with a very skewed distribution. So yeah, again, I apologize for kind of again saying we don't have the data to answer that, but we don't, unfortunately, at this time. Yeah, but thank you. It's a great question. I apologize. I can't answer it better.
No, that's okay. Thank you.
There's a question in the chat from Madison Smith about how in immunology they discussed that helminth worms and how they correlate with less inflammatory allergic responses in human populations. Has your research group, or are you familiar with research in the populations that you've been talking about of relationships among inflammatory pathways, helminths, and body stature?
Yeah, I've seen that research. It's been kind of an overall fascination with mine, but not something we've been able to look at. However, given the importance of or potentially the evolutionary importance of parasites to this population, it may be something we'd want to take a look at in the future. Yeah, I think they're alluding to the hygiene hypothesis, the idea that or maybe they aren't, but I'll just babble on about it. The idea that our dearth of parasites may be one reason for the increased prevalence of allergies, say food allergies, which is this fascinating thing. The immune system is so cool, and I'm not an immunologist, but I'm slowly kind of trying to become one based on this work. But it is a nifty hypothesis that there could be adaptation to a particular suite of pathogens, and maybe including European population in the USA or other US populations. There could be maybe mismatches between our current meager parasite load and allergies. Yeah, it's fascinating, but unfortunately, I don't have. It would be a very cool avenue of future research for sure. And there's a ton of weird stuff about parasite impacts on host genetics and parasite hijacking of the host immune system. And we've seen some hints of that in some other studies we've been doing, but it's something I would love to look into further. Yeah.
And
thanks for the question.
Yeah, Madison did affirm that she was talking about
oh, sweet.
The hygiene hypothesis. So
yeah.
spot on in your response.